Hello everyone, welcome to another lecture for drug delivery engineering and principles. Let us do a quick recap of what we done in the last class. So, in the last class we majorly talked about one major topic, which is immuno-isolated cell therapy, and this is in the immune module that we are currently at. And here we were looking for delivery of cells. And when I say delivery of cells, so a drug in this case is cells. Our actual drug is actually something else that is being produced by the cells. So, this could be insulin, this could be dystrophin. Insulin is of course if you are suffering from diabetes, dystrophin is for muscular dystrophy, or this could be something else also. I mean, these are the two major applications here, but you can potentially look for any disease that requires some protein as intervention, and you can then deliver cells that are producing those proteins. So, that is what we will discuss. So, in terms of delivery to cells, we had talked about that certain parameters, because most of the time the cells you will get will be from some foreign source, and there could be, so essentially any source of cells could be divided into three categories. One is autogenic, second is allogenic, and the third is xenogenic. Where autogenic represents something that is derived from your own body. So, if you are using autogenic cells, you do not really need to worry about immune system a whole lot, because your immune system will recognize it as your self cells, your self protein. So, it is not going to attack it. So, these things survive much better, but the problem is the source of these cells is fairly low, and in most cases are not there. So, I mean if you are suffering from diabetes, your own pancreatic cells are not working. So, that is why you need more cells from some different source. So, autogenic will not work in that case. Similar with the muscular dystrophy as well, where your proteins are not in the correct conformation or not in the correct orientation, and so your own cells will not work. So, you will need cells from a different source which have a functional copy, and so in that case allogenic is the most preferred, which means that getting it from same species. So, if since we are humans, we are talking about getting it from another human donor. So, in this case the immune system will act against it, because the immune system will recognize this as a foreign cell, and that needs to be protected. And of course, xenogenic is even more stringent in terms of immune response, because this is from a different species. So, this could be that let us say I get organs or cells from a pig and then try to implant that in my body, but obviously those are so different from my own body cells that the immune system will immediately recognize them and try to kill those cells. So, these were some of the sources. So, of course now because we are saying that mainly the allogenic and xenogenic is the source that we have, we need to protect these cells before we put them in the body, because otherwise the immune system is going to kill it. So, what is done in this case is you take a compartment which has a semi permeable membrane or some membrane, you put your cells within this compartment, the cells you are delivering and trying to protect from the immune system, and this membrane is such that it will allow small molecules such as glucose, oxygen to pass through, whereas big larger molecules such as antibodies or other immune cells are not able to go through this. So, that is the whole concept behind it, and we discussed several different conformation of these implants and all, but eventually this is what you want. Okay, so, let us take the discussion forward. So, let us talk about what kind of polymeric implants compatibility we need in this scenario. So, first is of course, and we have discussed a bit of it, is biocompatibility, which means that it should not really absorb a lot of serum and tissue proteins, and that is a bit obvious requirement, because let us say if this is your implant carrying your cell, if a lot of protein starts coating on it, it may first of all impede cell addition, the cell transport of different nutrients, as well as it can also lead to attraction of more immune cells coming in. As we discussed, protein adsorption typically is a layer which helps in more cells to come in and attach to that site. So, it is hence required that we completely characterize the type and amount of protein that are adsorbing on it, and we can use hydrophilic polymers. As we discussed, hydrophilic polymers are more similar to the environment that proteins are currently in, which is hydrophilic water. So, they will have less adsorption as well as conformation change, and sometimes even if you can't really work with hydrophilic polymers, what you can do is on your implant you can decorate it with our favorite molecule here, which is PEG, and what PEG will do is it will act as a windshield wiper, as we have discussed several times in this course, and make sure that none of the proteins are coming and adsorbing, even if this is a hydrophobic surface. So, those are few of the conditions and requirements in terms of biocompatibility. Then what about inflammation? So, it should reduce inflammation and cell adhesion at the polymer surface. So, it goes hand in hand with the protein data we already talked about, but even then it should reduce inflammation as well. So, so that will allow the barrier to not being formed in terms of diffusion of therapeutics and nutrients in and out, and success is essentially for these implants directly related to how much you can minimize the fibrous capsule, because eventually if your capsule is good and the body can't degrade it, then and if the body does not like it, then what it will try to do, it will try to wall it off, as we discussed. So, the body will first try to remove it. If it can't, then it will just wall it off, so that the rest of the body is protected from this particular capsule, but this is not what we want in our therapy, because we want it to continue to interact with various kinds of nutrients, with oxygen, as well as release of the product that we want from this particular device. So, the whole success for this will depend whether a fibrous capsule is formed or not. So, again if a fibrous thick capsule is formed, then that is the end of the device, because at that point you can't really have any kind of exchange happening at a substantial rate which you may need from this device. And again goes back to the requirement that you should have some sort of a hydrophilic polymer or at least hydrophilic coating. You can use PEG to do that. Typically uncharged surfaces will have less protein adsorption, so you can reduce the charge of your surface of these devices. And then finally we talk about immunocompatibility, and that means that the first of all antibody should not be able to penetrate, and we have already discussed this in the previous class, that the pore size should be such that antibodies are not able to go through, because if the antibodies go through, then they will impair the function of these cells. The second thing is we should reduce the complement activation. So, now this is very tricky, because in terms of most of the other immune system based molecules and cells, they are fairly large. I mean we are talking about antibodies which are more than 100 kDa, we are talking about cells that are in microns. So, it is easy to not have them penetrate and small molecules penetrate for glucose and oxygen. But when we talk about these complement proteins, as we discussed in couple of classes ago, that these complement proteins are also fairly small. So, we are talking about 10 to 20 kDa complement proteins floating around. So, it is becomes extremely hard to prevent complement activation from these surfaces, but there are different strategies that we talked about such as inactivating C5a, coating this with some molecules that may not let this complement activate. So, those strategies can be adopted in this scenario. So, that is why you need a very tight control over pore size to minimize these complement proteins as much as you can and still have a good flow of nutrients across this membrane. And not only size, but the charge, the secondary structure, all of this will influence the protein penetration through this membrane. And you also want to reduce cytokine penetration, because these are also small proteins just like complements that can go through. Again some of them are at least 10 to 50 kDa. which then you want to prevent, and of course you also want to have enough pore size for the glucose and insulin or whatever it is that these cells are producing to come out. So it becomes quite challenging, and that is why there is a limit to how much these implants last. Typically when patients get these implants survive for maybe 1 or 2 years, but after that within a 5-10 year survival is fairly low, and so you will have to get another implant, and so that puts more pressure on more organ donation, which is anyways quite low at this point. So these are some things to consider in mind when you are designing your vehicles for cell delivery. Okay, so now we are going to talk about gene therapy, which is again very similar to what we just discussed, but a little bit twist to the cell delivery. So let us talk about first of all what is genes and what do they do. So what do you think genes are? So to be very simple, basic definition will be genes encode for proteins. So anything that is encoded, that information is in genes at this point, and proteins are synthesized based on what the genetic sequence is. So you can change the protein sequence on the basis of the genetic sequence. Proteins do most of the biological and chemical work in our body. Genes are basically just storage units which are storing the information so that every cell can make more and more proteins. And we have about 40,000 different genes in our system, and those then combine together to form about 1 lakh different proteins. So all fine and good. So what happens when a gene is missing and mutated, and we briefly discussed about it in some of the disease circumstances, but of course if a gene is missing, then that protein cannot be synthesized by the body, or if it is mutated, then the protein could be non-functional. So let us say there is a gene that codes for let us say lysine, and then there is another gene that codes for, or another sequence that codes for, let us say alanine. So if somehow the gene sequences got mutated, that the lysine gene sequence is now coding for alanine, then your eventual protein that will form will, instead of having lysine, will have alanine, and maybe this lysine was fairly important in the function. So now the proteins will lose their function, they might even change their structure because of few proteins. So then that leads to functional deficiencies, because maybe this protein is involved in important function, let us say it is insulin. So maybe insulin is such an important protein that if we do not have it, it is very difficult for a person to survive, and various other diseases. So then what we can do is to overcome that, we can then, like we talked about in the previous 2 classes, we can encapsulate cells to do gene therapy. And so cells are, we are genetically altering them to produce and secrete high levels of these proteins that are missing from the person. So in the previous case we were more talking about diabetes, and in that scenario we are saying the immune system destroyed the whole cell. So the gene was still there, but that cell completely got destroyed, so it is not getting produced. So now all you can do is you can now genetically modify these cells and then have them produce insulin, that is just one example. So this process generally uses some kind of a viral vector such as a lentiviral or retroviral, and these viral vectors are extremely efficient in delivering a protein or a gene inside the cell, and then have them integrate with the DNA and then have them produce several copies of that. So if you have a gene mutated, you can take your own cells, you can then mutate them through this lentiviral or retroviral process, and then have them get a stable transfection. So that is one way. The other way is you engineer these cells are encapsulated inside a polymer microcapsule and implanted in vivo, similar to what we discussed previously for the islet therapy. So once you have these cells, you can then encapsulate them and use them for delivery in our body, and then these cells will secrete whatever proteins you want for a prolonged period. If you want them to be retrievable, you can have them in some macro device which you can then just take out, and this way you can avoid delivery of plasmid DNA, which we will cover in our next module. So it is much easier in that regards, it is much long term solution rather than delivering a plasmid DNA, and again we will come back to plasmid DNA in 1 or 2 classes. And again, just like the previous case, it is a very similar system. So, again this is the same example, it is probably the same figure. So, you have alginate polymer and you have calcium chloride solution. So, alginate is a negatively charged polymer, and then calcium of course is a divalent cation. So, what it will do, it will form bond between 2 different chains carrying negative charges, and that is how these chains will form an hydrogel, and whatever was present in the alginate gel initially before touching the calcium chloride solution will get encapsulated within these chains. So, again very mild process, you can use all kinds of cells, you can use muscle myoblasts, you can use let us say if you are deficient of factor 9 or factor 8, this is involved in blood clotting mechanism. It causes a disease called hemophilia, in which if there is a small cut the patients will bleed out. So, you can use these kinds of cells, deliver them, and they are encapsulated in a matrix, so surrounded and protected by the immune system if they are not self. So, here is just some examples. So, these are the alginate microspheres. And as you can see, each of these microspheres is encapsulating several cells inside. This microsphere could be about a millimeter or in that size range, and it carries several cells, and you can get millions of these microspheres, and depending on how much is the requirement for a patient, you can put these cells in there, which this barrier then prevents it from attack by the immune system. So, here is some literature example of when this was used. So, first they did an implantation in an immuno-deficient mouse, which means that the mouse does not have a good immune system, and so let us see what you see. So, in this case they were using the hemophilia factor, which was absent in this mouse, and so what you see is basically 2 things that they are measuring. One is the FIX and another is ABTT in a nude hemophilic mouse, which when the nude mouse here means that the immune system is knocked out, no adaptive immune system. And then hemophilic basically means that they can't clot blood. So, if these mouse get an injury or something, those, even if it is a minor injury, those injuries could be potentially lethal just because the blood will not clot, and so they will remove all their blood in due time, and of course with the lack of blood they will die because there will not be enough oxygen going to brain and heart. Okay. So, so this was one. Then they measured this antigen, which is the H-fix antigen. This is nothing but is produced by the cells that they have implanted. So, this normally is not present in the nude mouse, but since now that you have implanted these cells, they are also, apart from producing these factors that are needed for reversing hemophilia, we are also producing other proteins. So, they are measuring one such protein which is called H-fix, and what they find is, and this is of course the unit is in time, and this is showing the protein amount, and so what they find is after implantation they see increase in this amount of antigen, this H-fix, that is present in the mouse system. So, that means that the cells have actually grafted and actually survived all the way up to 80 days, and this particular antigen is also being produced. So, cells are healthy. The implanted cells are doing what they are supposed to do along with other functions and other survival proteins that they are producing. So, this is great. This was done in an N of 7. So, 7 mouse were used for this, 7 mice were used for this, and these are the cells that were used. Then the other thing that they looked at was the APTT level, and what is this? This is you can consider this as a reference for clotting activity. So, this is showing these seconds. So, how much time does it take for the clot to form, and so you see in hemophilic mouse earlier it was fairly high, but once you put this you see that the time for clotting has decreased. So, that means that there are more clotting factors now in this mice blood, and they are able to form a clot so that means that the hemophilia is somewhat cured, they will not bleed out completely or immediately. And then again the same thing you see is up to 80 days, I think that is what the authors, or 77 days, that is what the authors were looking at for the length of the experiment. They continue to see these cells produce what was required. Now of course this is immunodeficient mouse, so you would expect these cells to be fairly easily surviving, just because an immunodeficient mouse has no adaptive immune system to attack these particular cells, they do not have any immune system. So what happens in an immunocompetent mouse? So immunocompetent mouse is fairly a healthy mouse, that is complete immune system is present. But it is still hemophilia in this mouse, and so now they do the same thing again, and what they see is the factor 10, the H fix I was talking about earlier. This initially of course before implantation there is no antigen for this factor 10 which is missing in this mouse, and as the time increases you see that this implantation has worked, and this some amount of this particular antigen that is now floating in the mouse circulation. But then as time increases beyond 7 days you start to see a big drop, and eventually by the time 15-20 days have passed you see complete removal of this particular antigen. What does that mean? That the now immune system is attacking these cells, and it took a bit of time for the immune system to start developing response against it, but once it did you see even with the microencapsulation strategies we are seeing that these cells are dying, and that is because there are several cytokines and clotting factor and complement factors that are not letting this cell to survive. So big problem, and the same thing you can see with the time for clotting. So initially it went down a bit, there is a little bit of variability, but then eventually it starts to go back up as soon as this level starts to go down. So that means that this mouse is returning back to hemophilia, it is going back to that 1/22 that was before the implantation, and that is obviously not good, and so this is the major challenge here. So if it is not working in a mouse system, it is unlikely that this is going to work in a human system, and this is what lots and lots of research has been going on to, even in an immunocompetent mouse, how to get this to work better. And then here is another example. So now in that particular assay they tried to figure out if there is antibody against this human protein, and what they do find is yes, as the time increases the antibody titer against this human protein that they put in a mouse has also increased. So even though that human protein was functional for few days, once the antibody starts to come in, it is causing the immune system to get further and further active against these particular cell, and eventually causing death of these cells. So you can see how these antibody titers are increasing over time. So before that we were saying up to 80 days, there was 77 days there was no problem. Now we are seeing that even at day 25 or 24 these cells are gone. So what about if we do not deliver a cell? So now we are saying that we are delivering these cells, this is what is causing all this immunogenicity, the cells produce several other proteins, maybe some of them are causing the immune response to get triggered at a higher level. So what if we still do the gene therapy but remove cell from the picture or cell delivery from the picture? So that means that we need some other cell. So in this case what about we use our own cell? So going back to your autologous concept, what if we use our own cells, somehow genetically modify them and in that way, and when we put it back, at least the whole cell itself is not immunogenic. Maybe a protein is, maybe it is not, but it is much higher chance that these autologous cells will survive a lot longer in our system and will not be as immunogenic as the other ones. So again, these genes can be to replace a missing or a defective gene. So if a gene is completely missing, we can try to add 1 or 2 copies of those genes, or if it is defective, we can try to do the same thing. We can repair a non-functional or non-malfunction gene. So maybe its genes are big. So genes can be anywhere in kilobases long. What if there is only a point mutation? Maybe there is one sequence, maybe let us say A has changed to G, ATGC is the genetic code, right, nucleotides. So it is essentially composed of one or the other nucleotides throughout its genome. So let us say a G has changed to A and everything else is the same in that gene. So why do we change everything? Why can't we just repair this A back to G, and that way we will have a normal functioning for that particular protein. And then the other case is then we produce the therapeutic protein that helps to treat the disease. So do one of these two things, produce this protein which will help in treatment of the disease, and they can also be then used to vaccinate the patient against an infection, and I will come to that in a moment, but let us process this information that I just given you one by one. Okay, so here is what we are talking about. So let us say here is your cell, you have your genome, so it is several chromosomes are there. In one of the chromosome you have a defective gene here. So one thing you can do is you can add a gene. So this gene can integrate into the genome at some other site as it is shown here, or can just lie outside the genome but in the nucleus separately. So those are two options in gene addition or replacement. So if you actually are talking about, so this is extra chromosomal, which means that it is outside the chromosome but it will still lead to protein production. This is chromosomal insertion of course, because now it has integrated with your rest of the genome. And then this is gene repair, where what you are doing is, let us say if a portion of your gene is not functional, so you have an incorrect segment, rest everything is correct, you come in and you then insert the correct segment by replacing the defective segment. So that is gene repair, and it sounds very futuristic, but it is actually fairly feasible, and we will talk about it in a moment as well. So here is your correct template and you can then put it. But then there are several barriers to this. I mean it's good on the cartoon that yeah we can do this, and there is quite a lot of enthusiasm about it, but then there are several barriers of how to do this. So first let us talk about some extracellular barriers. So one is the DNA that you are delivering, if it goes in the plasma, which is of course one of the major fluid in a body, the blood vessels, if the gene is let us say in here, the plasma contains several nucleases, and these are nothing but something that degrades any long nucleotide chains. So this will get degraded, and once it gets degraded it is no longer functional. So that is a big problem when you are delivering an gene. Then there is problem that these external DNA can get uptaken and cleared out by the reticuloendothelial system, which is nothing but your immune system, so RES. So your spleen and your other immune cells may able to clear this DNA out. So not everything goes to the site. Then the problem is, let us say if I only want the gene delivery to happen in pancreas, let us say if it is for islets or it is for something else, then it is hard for me to go to that location and target only that location, because these will go everywhere. They are largely ineffective by the oral route, that is of course because most of it is not going to be able to make it to your circulation, although it does work relatively okay for immunization. Then let us say if we are using some other route, that is mucosal route. So, something that comes in touch with mucosa, then these gene do not really move around, diffuse very well in mucous layers. So, you do not really get a good movement of these genes in those circumstances. So, those are all extracellular while travelling through the body, and then there are several intracellular barriers. So, let us say if it does able to, after all these barriers, some of it has been able to reach the target cell. So, once it reaches the target cell, the first is how it is going to first of all go inside the cell, because the cell membrane is not permeable to such large molecules, as well as DNA is also charged. So, it is not, DNA is quite negatively charged, it has several phosphate groups. So, you are not going to be able to diffuse through the cell membrane. So, then it has to be uptaken by some endocytosis method that we discussed previously. So, but then the problem is that these endocytosis method will end up going to endosomes. So, if it does go to endosome, then you have to worry about how do you escape out from those endosomes, that is a problem. Quite a lot of it gets degraded in the lysosome. So, eventually the endosomes will go into the lysosomes, and that itself contain a very harsh environment and lots of nucleases as well. So, that is not going to work. Let us say if somehow this thing comes out in the cytoplasm, then how stable it is in the cytoplasm, that is another challenge, because that is not an environment where the DNA was supposed to be. And then let us say if it is in cytoplasm and it needs to go to the nucleus, then how does it travel to the nucleus, because it is a fairly large molecule, it has fairly low diffusibility through the cytoplasm. And then the same question arises again, that when it reaches the nucleus, that also is surrounded by a membrane, how does the DNA go from outside the nucleus to inside the nucleus. So, another barrier is translocation to the nucleus. I guess it is already written here. So, how does the nucleus, how does it go to the nucleus, and then finally it is whether it goes to the right region in the nucleus where it can get expressed. So, there are so many barriers that this DNA will have to go through, and this is a good point where we will stop, and we will see how we can overcome these challenges in the next class. Thank you.